We'll wait for everybody to join in, then we'll begin. Can you people see the chat now? Or is it still inaccessible? Is the chat still inaccessible to all of you?
Sir, it's not accessible.
I just wrote an hello. I mean, I don't know if you people can see that. Tried changing the settings, but anyway, it didn't work out. We are still getting people who are joining in. We'll wait for some time and see. So how is the course going for all of you? No responses? Am I audible?
Yes, sir.
We'll wait for some more time before everybody joins in. Meanwhile, if you have any questions from week 2, you may ask. Week 2, we mainly discussed about the DNA damages, how they occur, and what are the repair mechanisms involved. Let's see, people are still joining. So any questions from week 2? Nothing, it appears. It's a live interaction session. I would expect all of you to respond. So I welcome all of you to the second session of the live interactive schedule of the course Genome Editing and Engineering, which is taken by Professor Utpal Bora from IIT Guwahati. Just to introduce you to myself, I am Sanket Mukherjee, a Prime Minister's Research Fellow from IIT Delhi. So I would like to ask you if there are any questions regarding week 2. If there are none, then we can begin. Are there any questions? Okay, then let's start. We will discuss about some questions. Uh, so let's see. So transversion, it's any mutation where a purine base is replaced with a pyrimidine base, or a pyrimidine is replaced with a purine. B, a purine base is replaced with another purine. C, a pyrimidine is replaced with another pyrimidine. Or D, a purine or pyrimidine base is deleted. Any answers?
A.
A purine base is replaced with a pyrimidine, or a pyrimidine is replaced with a purine. Okay, so what is purine based replaced with another purine called? A purine base replaced with another purine, or a pyrimidine based replaced with another pyrimidine is called? Any answers?
Transition.
Yeah, you are right. It's transition. And a purine base replaced with a pyrimidine, or a pyrimidine based replaced with a purine is called transversion. So what kind of mutations are these? What are these mutations called? What kind of mutations could happen? What are mutations? Anybody would like to answer? Mutations are changes to the DNA that happens mostly due to an error-prone replication machinery, or it could be due to other reasons as well. So what kind of mutations usually happen? They're either substitute. Huh?
Base mismatch.
Yeah, you're right. So base mismatch is one of the ways how I mean mismatch repair is I mean it is kind of a repair mechanism. So it could be a base mismatch ideally. I mean a mutation could be either a substitution, it could be an insertion, or it could be a deletion. So what kind of mutation you think has happened here? I mean in case of transversion?
Substitution.
Right. So, okay, you're right. Then purine or pyrimidine base is deleted. So what happens if a base is deleted? What is
The
a DNA? Huh?
reading frame shifts.
Okay, that is correct. But if we say a DNA, how is a DNA formed? I mean, what does it contain? It contains a base and a sugar and phosphate bond. So what if a base is removed from this composition? Then what will we get? If there is no base, but there is a sugar and phosphate bond there. Have you studied about this? Any answer? Yes, no? It's called an A basic site. So A purine site or A pyrimidine sites, it is also called like that. So it's an A basic site. If a base is removed, but the sugar and phosphate bonds are still there, still intact. So let's talk. Okay, this is, I think there's some error. Just give me a moment. Okay. I think a wrong PPT has been uploaded. I'll just stop sharing and I'll just give me a moment. Yeah. Just a moment. There has been some error while saving this. I'll just quickly get you all the questions. Just give me a moment. Let's save here. Yeah, I think I have retrieved it. Just wait for a minute. Yeah, is the screen visible now? Is the screen
Yes,
visible?
sir.
Okay.
Yes, sir.
So as we talked about it, let's talk about transitions and transversions. So transition is a substitution where a purine is substituted by another purine. So that is what we call as a transition. Uh, Similarly, a pyrimidine, if they are substituted by a pyrimidine, then we call it again as a transition. But if a purine is substituted with a pyrimidine or a pyrimidine is substituted with a purine, then we call it as transversions. So what do you think? What kind of substitutions are more common? Transitions or transversions? Transitions are more common. Transitions occur more than transversion. So is that clear to all of you? Okay, let's go to the next question. So the key function of DNA topoisomerases is A, to replicate DNA, B, to synthesize mRNA, C, to synthesize protein, D, to relax superhelical DNA. What would be the answer?
D.
B. Oh, D. B or D?
D.
Yeah, you're right. So it will be D, to relax superhelical DNA. That is what DNA topoisomerases do. So what are the enzymes which are involved in DNA topoisomerases? Do you know? They are mainly helicases and gyrases. So which enzyme replicates DNA then?
Polymerase.
Yes, polymerase. You're right. So it's a DNA polymerase. Which enzyme synthesizes mRNA?
RNA polymerase.
Right. Then which enzyme synthesizes protein? Any answer?
Ribosome.
You're right. Ribosomes. So ribosomes, they have the tRNA, which is helpful for synthesizing proteins. So let's see what DNA topoisomerases do. They will cut the supercoiled DNAs. They will cut them at various sites and then they will try to relax this DNA. So by cutting and adjusting, there will be a relaxed DNA in the end. So the supercoiled DNA becomes a relaxed DNA using DNA topoisomerases. Different types of DNA topoisomerases, they work differently. And during this process, sometimes there are errors also which are introduced. So let's go to question number 3. So deamination of adenine produces hypoxanthine, xanthine, uracil, or thymine. Any answers?
A.
Hypoxanthine, yes, you're right. You're right. So all these products are formed during deamination reaction. So adenine, it produces hypoxanthine. Xanthine is produced by which base?
Guanine.
You're right. Uracil,
Cytosine.
yeah, cytosine. Uracil is produced by cytosine, and thymine, how
Methyl
is thymine? Sorry, what's your answer?
Methyl cytosine.
Yeah, 5-methyl cytosine. You are right. So let's look at the deamination reaction. A deamination reaction, it usually involves removal of an amine group which is all highlighted in red, as you can see. The clarity is not that good, but anyway. So cytosine forms uracil, adenine forms hypoxanthine, guanine forms xanthine, and 5-methyl cytosine, it forms thymine. So let's go to the next question. So which of the following below is an endogenous DNA damaging factor? Replication errors, ionizing radiation, ultraviolet radiation, or environmental stresses? Any answer?
Replication errors.
Yes, you're right. You're right. These are endogenous DNA damaging factor. This is an endogenous DNA damaging factor. So what is an endogenous DNA damaging factor? Okay, what are these? Then
External factors.
yes, these are exogenous factors or external factors. So exogenous DNA damaging factors or external factors, whereas replication error is one of the endogenous DNA damaging factors, or we could say an intrinsic factor. It's present in the body itself. So let's look at some endogenous factors and exogenous factors. So replication errors being one of the most prevalent form of DNA damaging agent, in at least endogenously. So replication errors, DNA based mismatches, topoisomerase DNA complexes. As I said, some topoisomerases, when they function, they sometimes introduce errors. Then there is spontaneous base deamination. We looked at it. Then there are A-basic sites. A-basic sites are also produced. Do you know of due to which enzyme? Any answers? Okay, it is usually due to glycolases. Then there is oxidative DNA damage and DNA methylation. So how does oxidative DNA damage happen? It is due to, yes,
ROA.
ROS.
yes, you're right. It's due to ROS. So these are all endogenous factors. These all happen inside your body continuously. But there are some exogenous factors also, like ionizing radiation, ultraviolet radiations, alkylating agents, aromatic amines, polycyclic aromatic hydrocarbons, reactive electrophiles, toxins, or environmental stress. So these are all exogenous. These are not usually present in your body, but you may be exposed to these things. So let's see about question 5. Photoreaction repairs, UV-induced DNA damage, replication errors, interstrand crosslinks, or A-basic sites.
A.
UV-induced damage. You're right. So this is how photoreactivation process happens. So the UV inactivated organisms, they restore their function by repairing UV-induced DNA damages with the energy of near UV light and an enzyme called photolyase. So as you can see in the right, this is the enzyme photolyase. So with the help of near UV um, light, it will uh, essentially repair this damage. So UV photo lesions and alkylated bases, these are two types of DNA lesions that can be easily reversed in an error-free way in lower organisms and mammals through photolyase mediated photoreactivation of UV lesions. So is that clear to all of you? Is that clear? What could be the damage due to UV? What could be UV induced damages?
Thymine dimer.
Dimer.
Yes, thymine dimers or pyrimidine dimers. So as you call it. So let's see the next question. Bulky DNA lesions such as cyclobutane, pyrimidine dimers and 6,4 photoproducts are repaired by base excision repair, mismatch repair, nucleotide excision repair, or photoreactivation?
D.
D. Is it D? Okay, so the answer is C. It's not D. It's not D, it's C. So essentially, wait. Yeah, so nucleotide excision repair, they repair the bulky DNA lesions such as cyclobutane pyrimidine dimers and 6,4 photoproducts. They are repaired by nucleotide excision repair. Any questions regarding this? Is everybody clear? Yeah, there are no responses. It will not be. Just give me a moment. It will not be photoreactivation. Usually when bulky DNA lesions are there in your body, when they are present, then they are repaired using nucleotide excision repair. So let's talk about the different kind of DNA repair mechanisms. So one is base excision repair. So when does it happen? It could happen due to spontaneous deamination or addition of a base. Something like this could lead to a base excision repair. So what happens? There is a mismatched base. As you can see, G should have a C, instead it has a U. So what will happen? The DNA glycosylase, they will excise this uracil, they will remove this, and there will be a basic site. We talked about A basic site, and later this site will be repaired and the correct base will be inserted here. Another one is nucleotide excision repair. So what happens? Nucleotide excision repair could have two kind of mechanisms. One is global excision repair. Another one is transcription coupled repair. So transcription coupled repair is for active genes, so that their repair could happen like early spontaneously. So that's why when transcription happens, along with that, these kind of repairs are, these kind of damages are repaired. While global excision repair happens for all the entire genome. I mean, irrespective of whether it's a transcriptionally active gene or it's an inactive gene. Well, let's go to the next thing, mismatch repair. So these mismatch repairs, they fix short insertion and deletion loops within repetitive DNA sequence that have resulted from strand slippage events and base mismatches produced during DNA replication. So you know what is a strand slippage? Anybody? Yes or no would do. Okay, nobody's
No.
okay. So what happens when, let's say there are multiple repeats, CAG repeats. There are multiple repeats, let's say in the order of around, there are 36 repeats. So what would be its complementary strand? GTC, GTC, something like this. So while the polymerase is acting on these strands, what could happen? Since there is a repeat here, what could happen is some of these nucleotides, they might slip. So some GTCs are here, then again GTCs, then again GTCs. Then again, so could you see the process? What happened essentially? The polymerase, it slipped. Is it clear to everybody?
Yes.
So this strand will have, let's say, how many? It will be more than 36 GTC repeats, or it will be less than 36.
More than 36.
Right. So this is essentially what happens in case of Huntington's disease also, like one of the mechanisms. So this event is called replication slippage or strand slippage. So these kind of errors, so there is a mismatch between the positive strand and the negative strand. So these kind of mismatches are repaired using uh, mismatch repair mechanism. So question 7, xeroderma pigmentosum is a rare autosomal recessive genodermatosis caused by mutations in DNA replication mechanism, nucleotide excision repair mechanism, transcription or base excision repair mechanism. Any answers? Seems none of you are interested to answer. So what would be the answer? Nucleotide excision repair mechanisms. So what is xeroderma pigmentosum? It's a rare autosomal recessive disorder. It is caused by mutations in the nucleotide excision repair mechanism. So what happens essentially, you develop severe photosensitivity and it results in sunburns, skin pigmentation, and even tumor formation is, I mean, the chances of tumor formation, they increase. And occasionally progressive neurological deteriorations also happen along with this syndrome. So xeroderma pigmentosum, it's an autosomal recessive condition with a complete penetrance that can be caused by mutations in any of the 8 genes. So there are essentially XPA to XPG genes. And these genes are also involved in the nucleotide excision repair mechanisms. So when there is a defect in the 8th XP gene, nucleotide excision repair is not affected. Instead, these XP variants have problems replicating DNA containing UV induced damage. So that is all about xeroderma pigmentosum. Let's go to question number 8. In DNA photolyase, reduced flavin adenine dinucleotide FADH works as a catalytic cofactor, a light harvesting cofactor, a metal ion, or an antenna molecule. Any answers?
A.
Yeah, reduced flavin adenine dinucleotide works as a catalytic cofactor. We will see how it works. So whenever there is UV light, which is less than 300 nanometers, what it causes? It causes pyrimidine dimers, right? That we all know. So these reduced FADH, they work as a catalytic cofactor in case of photolysis, and it will transfer the energy to CPD. So this electron, it will have an extra electron. It will transfer it to these thymine dimers in this case, or any kind of pyrimidine dimers. And because of this electron, this bond will break. So is it clear? So it's essentially a cofactor. In absence of FADH, this reaction or this repair mechanism will not happen. I'm really sorry for the this should be not Bacillus thuringiensis, but anyway. Any questions? Okay, let's go to question number 9. What is usually the methyl donor in formation of 5-methyl cytosine? Is it methyl transferase? Is it S-adenosine methionine? Is it cysteine or DNMTs? Do any one of you know the answer to this question?
I guess B.
S-adenosine methionine, it is usually the donor, methyl donors, for most of the methylated bases that we see in DNA. So what does then methyl transferase do? What is it?
It's an enzyme.
It transfers the methyl group from.
Right, you're right. You're right. You're right. And what is DNMTs? Anybody knows about it?
DNA. Methyl.
DNMTs, DNA methyl transferases, nothing else. It's the same thing. So essentially option A and D is same. So there are DNMT1, DNMT2, 3, so on. So these are all the enzymes that transfer the methyl group to the DNA. So essentially that's why it's called DNA methyl transferases. So let's go to the next question. Pyrimidine dimers are mainly formed due to UVA, UVB, UVC or X-rays.
UVB.
B.
UVC.
Yes, yes. UVC, not so much. UVC is even more damaging. So essentially UV induced damage, it could happen due to UVA or UVB. So DNA photoproducts are mostly formed, like pyrimidine dimers are mostly formed due to UVB, while UVA causes an increase in oxidative stress or immunosuppression. So here also some products are formed. They are like 8-oxoguanine. So 8-oxoguanine is a product of UVA induced damage. So this will result in mutations, and then it will, progressively there could be a development of cancer. So question 11, A basic sites in a DNA are repaired through nucleotide excision repair, mismatch repair, base excision repair, or photoreactivation. So all of you are clear what what is a basic site? Is it clear to all of you? Yeah, okay. Then which kind of repair mechanism will repair this kind of a damage?
A or B.
It's actually base excision repair. So in the process of base excision repair, there is production of an A basic site. This happens due to glycosylases. So this kind of damage, if it occurs in DNA, then it is repaired through base excision repair. So it happens in the process of base excision repair, and hence base excision repair is how it is repaired. So any questions so far?
I thought even nucleotide excision repair can be used to repair.
Yeah, nucleoside. I mean, yeah, there is essentially from in between base excision repair and nucleotide excision repair, it happens in two different phases. But if a base remains in the DNA, then a base excision repair, it will usually repair this kind of a damage. So these repair mechanisms are very, very much similar to each other. I mean, it's not like a particular damage is done to the DNA and only one sort of repair mechanism will be in place. So these are just to increase the proofreading activity of of your polymerases essentially. I mean, your DNA, it remains error free That is the idea. So any questions so far? Anything from the lectures? It appears there is nothing. So we will just go back to all the questions from the beginning and just discuss everything again. And then we will call it a session then. So the first question was, what is transversion? I mean, whether it's a purine replaced with a pyrimidine, or a pyrimidine replaced with a purine, or it is a purine base replaced with another purine, or it's a pyrimidine replaced with another pyrimidine, or it's a purine or pyrimidine base being deleted. So transversion is when a purine base is replaced with a pyrimidine, or a pyrimidine is replaced with a purine. So I hope you all know about purines and pyrimidines. So adenine and guanine, these are purines, whereas cytosine and thymine, they are pyrimidines. So any observation that you have between purines and pyrimidines? There is one cyclic ring here, and there are two cyclic rings in purines. So in pyrimidines, there is only one cyclic ring. So a transition would be from adenine to guanine, or guanine to adenine, and or a cytosine to thymine, or thymine to cytosine. Whereas transversions would be adenine to cytosine, or cytosine to adenine, or guanine to thymine, or thymine to guanine. It could also be C to G, cytosine to guanine, or guanine to cytosine. So these are all transversions. Then we wanted to discuss about the key function of DNA topoisomerases. So whether it is to replicate DNA, whether it is to synthesize mRNA, synthesize protein, or to relax superhelical DNA. So the answer is to relax superhelical DNA. That is the function of DNA topoisomerases. So this is how it works. So there are different types of DNA topoisomerases, but the end product is essentially a supercoiled DNA is converted into a relaxed DNA product. So deamination of adenine, as most of you answered, it causes production of hypoxanthine, while guanine, it causes production of xanthine. Cytosine, it causes uracil, and 5-methyl cytosine causes formation of thymine. So next we have which of the following below is an endogenous DNA damaging factor? So most of you answered it's replication error, while the rest three, ionizing radiation, UV radiation, and environmental stresses, these are exogenous DNA damaging factors. Now, talking about endogenous factors, replication errors, DNA-based mismatches, topoisomerase DNA complexes, spontaneous base deamination, A-basic site, oxidative DNA damage, which happens due to reactive oxygen species, and DNA methylation. These are all endogenous factors. While ionizing radiation, UV radiation, alkylating agents, aromatic amines, polycyclic aromatic hydrocarbons, reactive electrophiles, toxins, and environmental stressors, these are exogenous factors. Now, talking about photoreactivation, it's a UV-induced DNA. I mean, photoreactivation, it repairs a UV-induced DNA damage or replication error or interstrand crosslink or an abasic site. So photoreactivation, it usually repairs UV-induced DNA damage. Then we talked about the process of photoreactivation. So photoreactivation is the process by which UV inactivated organisms, they restore their function by repairing UV-induced DNA damages with the energy of near UV light and an enzyme called photolyase. So these UV photolesions and alkylated bases, these are the two types of DNA lesions that can be easily reversed in an error-free way in lower organisms and mammals through photolyase mediated photoreactivation of UV lesions. So question 6 was bulky DNA lesions such as cyclobutane pyrimidine dimers and 6,4 photo products are repaired by base excision repair or mismatch repair or nucleotide excision repair or photoreactivation? So the answer to this would be that bulky DNA lesions such as cyclobutane pyrimidine dimers and 6,4 photoproducts, they are caused by UV radiation and they are repaired through nucleotide excision repair. So we discussed about various kind of DNA repair mechanisms, DNA damage repair mechanisms, which included base excision, nucleotide excision repair, and mismatch repair. So we talked about xeroderma pigmentosum. It's a rare autosomal recessive disorder which is caused by mutation in the nucleotide excision repair mechanism. So as we said, XPA to XPG, 7 genes, these are involved in nucleotide excision repair essentially. Then talking about DNA photolyase, so reduced flavin adenine dinucleotide, it works as a catalytic cofactor. So we have seen how does the repair of repair happens. So whenever there is a near UV light, the electron is transferred from FADH to the thymine dimers. So this will cause the bonds to break and essentially we will get normal thymines. So this is how the damage is repaired. So the methyl donor, in case of 5-methyl cytosine or most of the methylated bases, it's essentially S-adenosine methionine. And pyrimidine dimers, they are mainly formed due to UVB rays, and we have seen how that happens. And then we talked about A-basic sites being repaired through base excision repairs. So any questions so far? Anything from the slides? I think I can stop sharing now. There are no questions then? So should we end this session if it is okay with you? Or are there any more questions that you would like to address? Okay. Seems most of you are fine. Anything else to discuss? Yeah, okay.
Can I ask something?
Yeah, sure.
So I'm just wondering, are you providing the recording of the sessions on the portal? In
We
case
we are recording these sessions and we are uploading it to our YouTube channels. I'm not sure whether it will be provided to you. So you
Okay.
can definitely go through at least my recordings in my channel. So we have to upload this to NPTEL. So I'm not sure whether they will share this with you, but ideally this is done so that you guys can maybe catch up if there is any doubt.
Thank you.
So the
So I can
recordings are
just check.
there. Yeah,
So
you
it's
can
there
check.
on your portal. Like, yeah. Thank you.
Anything else? Any further questions? If there is nothing more, then you guys can leave. We can call it a session then. We will meet again on next Tuesday on 6 p.m. right at this time. Is it okay? Okay.